Hi everyone, let's see the carbohydrate lab protocol. We have two experimental parts. In the first part, we are investigating the browning reactions of sugars. As you know, under some conditions, reducing sugars produce brown colors that are desirable and important in some foods. At other times, brown colors obtained upon heating or during long-term storage of foods containing reducing sugars are desirable. We have two procedures in the first part. First procedure describes the preparation of caramel sauce prepared at three different temperatures, which are 220, 240 and 260 degrees Celsius. And here are the ingredients. We have sugar, butter, half and half cream and vanilla extract. To get started, we place sugar in a saucepan, then turn the heat to different levels. And heat the sugar by stirring until we see a rich brown color. Then we add butter and cream. So first browning reaction occurs, then we add butter and cream. Then we measure the color using a Hunter LAB colorimeter. So in the second procedure, we are investigating another browning reaction. Here we have glucose and amino acid lysine. And they are reacted at three different pH levels: acidic, neutral and pH, in the presence of heat. After reaction, the mixtures are transferred into a cuvette and absorbance values are obtained. Remember, we use spectrophotometer for measuring absorbance. It measures the intensity of light passing through a sample solution in a cuvette and compares it to the intensity of the light before it passes through the sample. So the measurement is based on the interaction of sample with light. And we know that the amount of absorbed light is related to the amount of sample. There is a linear relationship between absorbed light and concentration. Here we want to measure the intensity of the brown color. So that means the greater the absorbance reading, the greater the brown color, the intensity of brown color, or the concentration of the brown color. And here are the results. We have LAB values for different cooking temperatures for procedure 1, and we have absorbance readings at three different pH levels for procedure 2. So in your assignment, you need to discuss reaction mechanisms for brown color formation. Also here, B plus B represents the yellow color. We also see some yellow color formation at this temperature. You can also discuss that. So procedure 1 and procedure 2 have different reaction mechanisms, okay? You need to notice that. The first one, we have half and half cream, we have amino acids, but they are not reacting because we are adding half and half cream after browning of sugar takes place. So they are both non-enzymatic browning, but the reaction mechanisms will be different. And in procedure 1, you need to comment what's the effect of heat on color of caramel sauce. So discuss what factors contribute to the browning, which reactions involved in both cases. What are the chemical changes that take place when heating sugar only or heating sugar with amino acids, okay? You can talk about the aroma, flavor. And also, what's the effect of pH on color formation in procedure 2? here we have at alkaline conditions, we have better reaction rate. So what's the reason for that? Okay. So you need to discuss that points in your assignment. Let's look at the second part. And the second part is about the extraction and quantification of pectin. So what is pectin? Pectin is a polysaccharide. It is heteropolymer. It's the component of primary cell wall with D-galacturonic acid, with some of the carboxyl group esterified with methanol. So pectin is responsible for the texture of fruits and vegetables. So for example, when fruit ripens further, its pectin content decreases, resulting in a loss of firmness. And it's commercially prepared mostly from some byproducts of the food industry, such as apple pulp, citrus pulp, and sugar beet pulp. So in this experiment, we extract pectin from apples in the presence of heat and acid. We are adding lemon juice, so it's a citric acid. So we need that citric acid to break down the pectin. I mean, to extract pectin by breaking down the apple cell wall. Then we quantify lignin using a wet chemical method, and on the other hand, we also use that pectin to make a jelly. Because pectin is capable of forming gels in aqueous systems. If we have a high contents of solids and low pH values, pectin form gels. So we are adding sugar. Also, there is some acidity coming from grape juice. So what is the function of pectin in jelly making? So high soluble solid content, which is when we have sugar, and you know it's soluble in water, so we will have high soluble solid content in water, and it causes dehydration of pectin by competing for water through hydrogen bonding. And the low pH suppresses ionization. So because of that dehydration effect and charge effect, ionization, pectin forms gel. Okay? This high soluble solid content causes dehydration of pectin by competing for water, and it happens through hydrogen bonding when we have sugar in water, high concentration of sugar in water. And low pH, the acidity coming from grape juice, suppresses ionization. So the gel is formed by charge and dehydration effect. That's the basic mechanism. Let's see the other results. So here is the data to calculate the pectin extracted from apples. We have galacturonic acid standard curve here. You are going to use that. What's the x-axis and what's the y-axis? On y we have the absorbance differences at 450 minus 400 nanometer, and x-axis is the galacturonic acid. So remember, we said here pectin is the component of primary cell wall with D-galacturonic acid. So that's why here this galacturonic acid concentration will represent our pectin amount. So for our calculation, we are going to use that. Okay. So first you need to subtract the absorbance at 4 nanometer from the absorbance at 450 nanometer, these two. Then this will go to that equation. It will be your y value. Then you are going to solve this equation to find x. And x will be the galacturonic acid, which is your pectin amount. So it will be in millimole per milliliter. So you have the molecular weight of galacturonic acid given here. You can use that to make your conversion. And you know the total volume of the pectin extracted at the first place. So with this given data, you can calculate the amount of pectin extracted from the apples. So that's all for this lab. Let me know if you have any questions.